So, good morning, good afternoon, and good evening to everybody. Welcome to everyone to today's Global Immunotalk. Global Immunotalks are a wonderful initiative created by Dr. Carla Rotlin and Elena Sunigan in 2020 to spread immunology and inspire the new generation of immunologists all around the world in a friendly and egalitarian manner. I am Gabriel Rabinovich and have the honor and privilege of presenting our today's speaker, Dr. Padmanee Sharma. Dr. Sharma is an internationally renowned physician scientist at MD Anderson Cancer Center, where she serves as a professor in genitourinary medical oncology and immunology, associate VP of immunobiology, and holds the TC and Janet Su and Doe Chair in Cell Biology. She is the inaugural scientific director of the immunotherapy platform at MD Anderson, overseeing immune monitoring studies for over 100 clinical trials. In 2022, she was appointed the Director of Scientific Programs to the Jim Allison Institute. Her pioneering work includes leading the first neoadjuvant trial with immune checkpoint therapy. She has also identified multiple mechanisms of response and resistance to immune checkpoint therapy. Her work also led to the development of multiple innovative immunotherapy clinical trials, including trials that led to the FDA approval of immune checkpoint agents to treat bladder and renal cell carcinoma. She has received numerous prestigious awards, including the Emil III Award, the Cole Award, and the Randall Prize. Dr. Sharma is a member of the American Society of Clinical Investigation, member of the American Association of Physicians, and fellow of the Academy of the American Association for Cancer Research. In addition to her research, Dr. Sharma is a dedicated mentor. She has guided over 50 students, fellows, and junior faculty since 2005. Dr. Sharma earned her MD and PhD in immunology from Pennsylvania State University in 1998, completed her residency at Cornell Medical Center in 2000, and her postdoctoral work plus fellowship at Memorial Sloan Kettering Cancer Center in 2004. In a personal note, Pam is one of my favorite scientists. She's a pioneer in the field of clinical immuno-oncology, and that's great, either in basic, translational, or clinical immuno-oncology and back again. Additionally, Dr. Sharma is one of the most generous scientists I have ever met. So thank you very much, Pam, for sharing this session and for sharing your work. The title of Dr. Sharma's talk today is From the Clinic to the Lab, Investigating Mechanisms of Response and Resistance to Immune Checkpoint Therapy. So Pam, as you know, one of the traditions of Global Immunotalks is to ask a special question before you start your talk to inspire the young generation of scientists. So the question today for you is, what advice would you give to early career scientists about building a strong professional network?
I think for the young scientists out there, it's so important to think about networking with people in teams, to have a collaborative spirit so that you work with people and you're generous with people, but also to remember that mentorship is important. So selecting senior people who can help to mentor you, even if they're not at your institution, they could be elsewhere in the world, because we can be so easily connected across the world these days. It's important to have that senior mentorship to help guide you, have conversations with, and help open doors for you as you move through into your career.
So thank you very much, Pam. Thanks for the very inspirational words. Now I would like to invite you to share the screen and deliver your lecture. Thanks so much.
Thank you, Gabby, and thank you so much for that kind introduction. I'm going to, hopefully you can see my screen now. Let me just put it in presentation mode. So I'm going to speak today about the work we do between the clinic and the lab and going back and forth. I changed the title slightly because I call this process of going between the clinic and the lab reverse translational approaches. And I hope that you'll see some of that as I speak today and take you through the work that we've done over many years, as Gabby's pointed out. These are my disclosures listed here. So I just want to remind everyone that the reason that we've had so much success in the clinic with immune checkpoint therapy is because it came from basic science. It really came from understanding the mechanisms that drive T cell responses. And so without understanding the basic biology and the basic pathways that influence T cell responses, we were never going to get to success in the clinic. So the intrinsic signals that regulate a T cell response is super important to understand. And as you may all be well familiar with, there are two signals. The T cell receptor is signal 1 and CD28 is signal 2. These same two signals are very important to turn a T cell on, but it's exactly the same two signals that also regulate inhibitory pathways such as CTLA4. And this really highlights this yin and yang to the immune response. So you can drive it in one direction and it will always try to come back to controlling itself and having a homeostatic mechanism. So by understanding this, we moved away from trying to turn T cells on with vaccines, and really then Jim Allison proposed the blockade of the inhibitory pathway on T cells, blockade of CTLA4 was going to be the way to actually turn on anti-tumor T cell responses. So this was really the paradigm shift and the beginning of the entire field of immune checkpoint therapy. And clearly, it led to lots of success in the clinic. Here's a patient who had stage 4 metastatic melanoma with multiple tumors that can be seen on the CT scan shown here, and then received anti CTLA4 therapy, and all of the tumors went away. So, about 20% of stage 4 cancer patients have durable responses, and a subset of patients are even considered cured. 20 years later, there's still no recurrence of disease, and these patients are doing well. This led to the approval of anti-CTLA4 in 2011. And anti-CTLA4 is only one of multiple immune checkpoint therapies being studied, right? This is the new field now that we're dealing with. PD-1 and PD-L1 is another approved antibodies that are in clinic right now, but we still don't have a full understanding of all of these immune checkpoints and how they're playing a role. So the basic science still needs to drive our understanding so that we can take advantage of that in order to translate these into the clinic. So there's still a lot of work going on the targets that are highlighted in this cartoon, but even this cartoon diagram is outdated in terms of the number of targets that are being investigated. So I just want to point out again that even with anti-PD-1, this is a study that we led in patients with metastatic bladder cancer. You can see each of these bars represent a patient, and when the bars are going down, this is when the tumors are regressing, and you can see there's a great proportion of patients whose tumors decrease in size and the patients do better. However, there are clearly patients where the tumors are increasing in size, and so we still don't understand why some patients are responding and others are not. Based on the data, though, with immune checkpoint therapy, we've had multiple FDA approvals for immune checkpoint therapy, as shown here, across many, many different tumor types. And really, the important thing to take away is because immune checkpoint therapy is not targeting a specific tumor cell. The immune checkpoint therapy is targeting the immune system, and then the immune system can then help to eradicate tumor cells wherever they may be and wherever they originate from. And this is very important because now we even have FDA approvals for genomic alterations such as MSI status and tumor mutational burden. So the key research questions that remain, of course, are why do some patients respond and others do not? Can we identify biomarkers that predict response or immune-related toxicities, which is another important issue to discuss because these toxicities with the immune checkpoint therapy agents are very different from toxicities that we see with chemotherapy or radiation therapy. Are there biomarkers to enable patient selection for treatment monotherapy, such as anti-CTLA4 alone or anti-PD-1 alone, versus combination therapy of anti-CTLA4 plus anti-PD-1? The combination therapy has better efficacy, but it also has higher rates of toxicity. So these are things that we need to distinguish which patients can get a monotherapy and which patients should get combination. Are there other pathways that can be targeted to improve clinical outcomes, and can we increase the number of patients who respond? And so, to try and address some of these questions, we adopted this reverse translational approach. I mean, most of you are aware that in the laboratory, hypothesis testing is best done in the mouse models. I mean, mice are inbred, the disease is homogeneous. It's very easy to knock in a pathway or knock out a pathway and get an understanding of how that pathway is operating. However, patients are polymorphic and the disease is heterogeneous. So, in patients, the most we're going to get is correlative data. And that correlative data can then lead to a generation of a hypothesis. And then we must take that hypothesis back to the laboratory from the clinic, and then test the hypothesis in an immunocompetent model in the lab. And the reason this becomes important is because at this point in time, we have about 3,000 clinical trials ongoing with immune checkpoint therapy agents. And that's a lot of clinical trials that are ongoing. And we've, at this point, have the clinic outstripping the basic science understanding. So we have to find a way to use the clinical information, take it back to the lab, and really advance our knowledge and our understanding using that reverse translational model. So I'll give you some examples of how we're doing that. One of the things that we had to do was rethink clinical trial design in order to do this. As you're aware, most clinical trials are phase 1 safety dose escalation, phase 2 efficacy, or phase 3 comparison to standard of care. We proposed that we could utilize these pre-surgical or tissue-based trials, which are called phase 1A or phase 2A studies. We designed this in a neoadjuvant setting, meaning patients with localized disease before they go to surgery, whereby we can give treatment and then get all of the tumor samples at the time of surgery to understand what the human immune response is doing within the tumor microenvironment. So these studies allowed us to gain insights into clinical signals for tumor types that were not being studied previously, and also gave us the ability to look at biomarkers and gain mechanistic insights into the pathways that are at play as a response to therapy. We initiated these studies in 2004, and the first trial was in 2006. I highlight these dates because this was all prior to any FDA approval of an immune checkpoint therapy agent, which first occurred in 2011. And so I encourage the young people to think about this as we're doing so many different clinical studies right now with new agents. And we may not always understand the mechanisms of these new agents, and they're just going into these stage 4 metastatic disease setting. Maybe we need to do small clinical trials to understand their mechanisms in the human immune response before we can propose how to then move them forward in the larger clinical trials. So this was the first neoadjuvant immune checkpoint therapy clinical trial we conducted. This was in patients with localized bladder cancer. This was also the first clinical trial in patients with bladder cancer for immune checkpoint therapy. And you can see that it was a small trial. It was 12 patients. And the reason it's 12 patients is because these are patients I see in my clinic. So as a medical oncologist in the GU medical oncology field, I can enroll patients onto my own clinical trial and then take the samples back to my laboratory. And so by writing this clinical trial and enrolling patients, we were then able to collect the samples at the time of surgery. And so the clinical trial really gave us multiple pieces of information. One, it let us know that it was safe to give immune checkpoint therapy prior to surgery. We established the safety, and that means that neoadjuvant clinical trials are now many, many more ongoing with immune checkpoint therapy. It also gave us a clinical signal for efficacy because we saw that 3 patients developed pathologic complete responses. And so this indicated to us that bladder cancer can be responsive to immune checkpoint therapy. And therefore, we then did the clinical trial in the setting of metastatic disease and obtained FDA approval with anti-PD-1 nivolumab for patients with bladder cancer. Importantly, though, the clinical trial also led us to design new clinical trials in the neoadjuvant space because we had that safety data. And I just want to point out that we were also able to do this combination clinical trial with anti-CTLA-4 tremelimumab and anti-PD-L1 durvalumab as a combination study in patients with localized bladder cancer who are ineligible for chemotherapy with cisplatin. And so these patients do not have a treatment prior to surgery, and they have very high risk of disease recurrence at a later time. And so we gave them this combination therapy study, and we could see in this study that these patients had pathologic complete responses of 37.5%. And even in patients with large, bulky disease, who was unclear that they would even make it to surgery, they were able to complete the surgery. And even in that group of patients, we saw pathologic complete responses of 42%. Again, highlighting the importance of the neoadjuvant setting for having clinical impact. Importantly, again, though, we were able to study all of the tumor tissues that were obtained from these clinical trials. And so we can look at the pre-treatment and post-treatment samples and ask what are the important things that are happening. And again, highlighting that this was done in 2008. So at that time, we didn't have the great technology that we have now in terms of single-cell RNA sequencing, CODEX, CyTOF. All of those technologies have really helped what we can do in human immune monitoring. But back then in 2008, I'm showing data where we did simple single staining immunohistochemistry. And you can see even in these simple studies, you can start to see that there's an increase in CD3, CD4, CD8 T cells. These are granzyme positive. And even at that time, we were able to appreciate these CD20 B cells that are also infiltrating into the tumor microenvironment. Again, giving an indication of how immune checkpoint therapy, and specifically anti-CTLA-4, was changing the tumor microenvironment and how the human immune response was changing over time. What we did see as we studied these tumor samples more from both of these clinical trials in patients with bladder cancer, we identified these tertiary lymphoid structures that were occurring in the tumor microenvironment. And for those of you who are familiar with tertiary lymphoid structures, they usually occur just within the lymph nodes. That's where they predominantly exist. And so for the first time now, we're reporting that tertiary lymphoid structures can form and be maintained within the tumor itself. And the patients who had these tertiary lymphoid structures, you can see in this red bar, these were the responder patients who had better outcomes. And these tertiary lymphoid structures were made up of CD4, CD8 T cells, dendritic cells, as you can see here. And the increase in the particular subset of T cells that we identified were these ICOS positive CD4 T cells. And so it was interesting to see that in the responder patient, these ICOS positive T cells were increasing, but not the non-responder patients. We then did codex analyses. Now that we have these technologies later on, we were able to then go back to our samples and perform codex analyses to try and identify what are the cell-cell interactions and what are the receptor ligand pairs that we can potentially look at for helping with the formation of these tertiary lymphoid structures. And here in just some preliminary data, we're showing that it's CD8 T cells, these ICOS positive CD4 T cells, and these BATF3 positive dendritic cells that seem to be forming the neighborhood for these tertiary lymphoid structures. And so these analyses are still ongoing at this time. But what is ICOS? What did we identify in terms of looking at this ICOS positive subset? So ICOS is inducible co-stimulator, and it belongs to the CD28 and CTLA-4 family. Its expression is increased on activated T cells, including regulatory T cells. So it's interesting that we would think if regulatory T cells were in the tumors, we would not see the good outcomes that we did in patients who had an increase in ICOS. So given this diverse role that was reported, we wondered what ICOS could be doing. And for those of you who are aware that within the lymph nodes, ICOS is mostly associated with follicular helper T cells, but their transcription factor at that point is BCL6 that helps to drive then these follicular helper T cells to help between T and B cell collaboration. But a role in anti-tumor responses had not been established, and so we wanted to explore this more. And so that was all of the clinical data that we had that I showed you. And so then we said, okay, what does the clinical data tell us? And so the first hypothesis we generated from the clinical data was that the ICOS ligand pathway is necessary for effective anti-tumor immune responses in the setting of anti-CTLA-4 therapy. And so we went back to the laboratory in mouse models, and we could test in wild-type mice, ICOS knockout mice, and ICOS ligand knockout mice, how they responded to treatment with anti-CTLA-4 after tumor inoculation. And you can see that the wild-type mice had much better tumor rejection and long-term survival compared to the ICOS knockout and ICOS ligand knockout mice, indicating an important role for the ICOS ligand pathway. We then wondered, why is the pathway necessary for optimal anti-tumor responses? And this led to the second hypothesis that we generated. As I pointed out, ICOS is related to CD28 and CTLA-4, and they all have this cytoplasmic PI3 kinase binding domain. And so we hypothesized that ICOS can induce signaling via PI3 kinase to promote Tbet expression, and Tbet is a transcription factor for Th1 responses, which are very important for tumor rejection. So we wanted to test this hypothesis, and again, we went back to the mouse models, and we could use wild-type mice and ICOS knockout mice as shown here. And then we took the CD4 T cells from these mice, and in the untreated setting, you can see neither the wild-type nor ICOS knockout mice have a lot of Tbet expression. However, after we treat the mice with anti-CTLA-4, you can see there's the wild-type CD4 T cells are now capable of increasing Tbet expression, but the ICOS knockout CD4 T cells are not increasing Tbet expression in the same way, either in the spleen, tumor draining lymph node, or in the tumor infiltrating lymphocytes that we looked at. So indicating again that ICOS was playing an important role for helping with Tbet expression. Even though all of these T cells have CD28 expression as well, you can see ICOS also plays a role here in prolonged having Tbet expression over time. But we wondered whether or not ICOS was signaling through the PI3 kinase pathway to allow for the Tbet expression. And to do those experiments, we used the ICOS YF mice, And these are mice that have a single amino acid change in the cytoplasmic tail from Y to F, so that it's no longer capable of signaling through the PI3 kinase pathway, but it still expresses ICOS. And you can see when we looked at these mice, both wild type and ICOS YF mice, you can see now it's clearly the ICOS high expressing T cells from the wild type mice that have the T-beta expression, whereas the ICOS YF mice fail to have T-beta expression in either the ICOS high or ICOS low T cells that we sorted. And so these mice also had impaired anti-tumor responses and tumor rejection. So clearly ICOS was an important pathway, and this led us to then the third hypothesis that the ICOS ligand pathway can be targeted and developed as a combination therapy strategy. And so we did these experiments in collaboration with Jim Allison's group, whereby we targeted both ICOS and gave anti-CTLA-4. And you can see in the wild type mice in the blue dotted lines that the combination therapy led to really improved anti-tumor responses and long-term survival. And in the manuscript that we published, we showed that this was due to increase in interferon gamma, TNF alpha producing CD8 and CD4 T cells. And in the ICOS knockout mice, when we did the experiments, we did not see this, clearly indicating the role for ICOS in this pathway. So now there are many companies developing ICOS targeting agents, either as bispecifics or single oncolytic virus models, and we're waiting to see how those clinical trials will go. We don't have data yet from that. But I do want to shift gears a little bit now, since we talked about melanoma, bladder cancer, lung cancer. These are tumor types that are very responsive to immune checkpoint therapy. And it's thought that they're responsive because they have lots of mutations within these tumors. And because they have lots of mutations, they have lots of antigens for T cells to recognize. And so when you give immune checkpoint therapy, it will work better in these tumor types. However, tumor types like prostate, glioblastoma, pancreatic cancer, it's thought that they have very few tumor mutations. And because they have very few tumor mutations, they don't have a lot of antigens for T cell recognition, and therefore they will not respond to immune checkpoint therapy. And so from a clinical standpoint, I agree with the statements, because in clinic, we've seen that the prostate cancer clinical trials have not been able to show reproducible clinical benefit for patients receiving immune checkpoint therapy. And so those clinical trials did not lead to any approvals for immune checkpoint therapy in patients with prostate cancer. And so we were wondering, though, as an immunologist, is it really true that it's because there aren't enough mutations? Because again, it should only take a single mutation, which prostate cancer has, for a T cell to recognize it. It doesn't need hundreds of mutations for a single T cell to recognize an antigen. So we wanted to understand if there were other resistance mechanisms within the tumor microenvironment. And so we conducted another neoadjuvant clinical trial. Again, look at the date, it's 2009, prior to any approval of an FDA agent. And this time we were asking patients who had localized prostate cancer and were scheduled for surgery to receive 2 doses of anti-CTLA-4 in a small trial of 20 patients, so that we can understand what was happening in the human immune responses within the tumor microenvironment to lead to resistance with anti-CTLA-4. And what I can say, shown here in this immunohistochemical study again, is that pre-treatment, there are very few infiltrating T cells in prostate cancer. So it really is a cold tumor microenvironment. However, after treatment with anti-CTLA-4, you convert this cold tumor microenvironment to a hot tumor microenvironment. So you can see increase in CD4 and CD8 T cells are shown here, even increases in ICOS positive and granzyme B positive T cells. So it was very interesting to us that we were converting this cold to hot tumor microenvironment, but we were still not getting clinical responses. But as I mentioned, the immune response is very tightly regulated. And when we looked at our gene expression studies, we saw not only were we increasing these effector responses, we were also increasing compensatory immune inhibitory pathways, including PD-L1 and VISTA, as shown here. And so when we went back to the immunohistochemical studies and compared pre and post-treatment samples, you can see pre-treatment that there really is no PD-1 or PD-L1 or VISTA in any of these tumors that we looked at prior to treatment. However, after treatment with anti-CTLA-4, now you can see PD-1 is increased on immune cells, PD-L1 is increased on immune cells and tumor cells, and VISTA is increased on immune cells. It's very important to recognize that a single time point of a tumor biopsy is not going to give you sufficient information to understand how the immune response changes longitudinally over time, because the immune response is so dynamic that a single time point really wouldn't give us all the information. So it's important that we had these different time points to look at, because now we can see how these inhibitory pathways are being increased within the tumor microenvironment. We asked where VISTA and PD-L1 were predominantly expressed within the tumor tissues, and we found that they were predominantly on CD68 macrophages. And when we asked whether or not it was the same CD68 macrophages that expressed both VISTA and PD-L1, or whether they're two distinct subset of immunosuppressive macrophages, we found in this multiplex immunofluorescence study that really the CD68 cells were either VISTA positive, which was in white, or PD-L1 positive, which is in pink. Very few cells were both VISTA and PD-L1 positive, indicating the likelihood of needing to block multiple immunosuppressive subsets in order to get clinical responses in prostate cancer. We knew that the VISTA and PD-L1 were previously published as potentially being immunosuppressive, and we wanted to test this in our studies. And so we took prostate cancer patients' T cells and incubated them in vitro with anti-CD3, and can show that these CD3 T cells could then produce interferon gamma and TNF alpha. However, if we coated the plate with PD-L1 Ig, VISTA Ig, or a combination of VISTA Ig and PD-L1 Ig, you could see this decrease then in interferon gamma and TNF alpha production from the T cells, indicating that these are really inhibitory pathways. And now there are clinical trials that are targeting VISTA and PD-1, and these are ongoing by different companies at this point. So we're waiting to see if that data will also lead to any clinical benefit. We took our data showing that anti-PD-1 and PD-L1, showing that PD-1 and PD-L1 were increased within the tumor tissues of patients receiving anti-CTLA-4. And we asked Bristol Myers Squibb to collaborate with us on a new clinical trial whereby we could give anti-CTLA-4 and anti-PD-1 in combination to patients with metastatic castration-resistant prostate cancer. And I won't go into all of the details because the study is published here. And I will say that with the combination study, we were able to see these types of responses where a patient has PSA of 500, becomes undetectable after treatment, and soft tissue diseases within the patient's CT scan also regressed after treatment. We were not able to continue the trial because of toxicities. And as I mentioned, toxicities do occur as you now move from monotherapy to combination therapy. But what we did realize was the patients who were responding to this combination therapy were patients with soft tissue metastases. And patients with bone metastases were not having the same type of clinical responses. And for those of you who know prostate cancer, you know that it's a disease where predominantly the metastatic site is the bone. So it was really interesting to us that we could see differences in what's happening within soft tissues versus bone metastases. And so we wanted to understand that better. And then we studied the soft tissue samples that we had collected from patients and the bone metastases that we collected from patients. And we could ask what was the difference that was happening in the immune response in the soft tissue lesions versus the bone metastases. And you can see here that patients with soft tissue metastases, when you looked at the pre-treatment and post-treatment samples, you see this significant increase in the Th1 interferon gamma response that happens within the soft tissue lesions after treatment with anti-CTLA-4 and anti-PD-1 after combination treatment. However, when you look at the bone metastases and look at the pre and post for Th1, you never see in each of these graphs as a different patient, and we never saw this increase in the Th1 response. And what we actually found was a significant increase in Th17 within the bone metastases. And so we wanted to model this back in mice to understand it a little better. And so we went back to a mouse model that we could then inject castration-resistant prostate cancer cells into the subcutaneous areas to mimic the soft tissue lesions, or into the bones of the mice to mimic bone metastases. And then we treated the mice with anti-CTLA-4 and anti-PD-1, very similar to the clinical trial. And again, in the bone lesions, we were able to show that the Th1 response never increases after treatment with the combination therapy, but the Th17 response increased similar to what we saw in the clinical setting. And then when we looked in the soft tissue or subcutaneous area, you can see again in the mice, there was a significant increase in Th1 responses, no increase, actually decrease in Th17, because we were now skewing the T cells in the bone lesions towards Th1 rather than Th17. So why were we getting the skewing in the bone lesions towards Th17 and not in the subcutaneous or soft tissue areas? And so we went back then to look more at Th17 biology. And for those of you who are aware, Th17 biology requires two cytokines, IL-6 and TGF-beta signaling at the same time to help drive Th17 responses. And when we looked in the bone marrow microenvironment, you can see the BM is bone marrow and BMT is bone marrow with a tumor. Regardless of whether there's a tumor, IL-6 levels are much higher in the bone compared to the serum. However, TGF-beta is only high when there's a tumor within the bone microenvironment. And we showed in the manuscript that was published, this was due to osteoclast activity. And so we proposed that in order to treat the bone metastases, we may need to block TGF-beta. And so we did a combination study in our mouse model of anti-CTLA-4, anti-PD-1, and anti-TGF-beta. And you can see now that in the green box shown here, for the first time, we were able to get decreases in the tumor volume as a result of combination therapy. Importantly, we also saw for the first time in the bone microenvironment, now we were able to have expansion of these Th1 CD4 T cells and also expansion of CD8 T cell clones within the bone microenvironment by blocking the TGF-beta pathway. And we also showed that as a result of this, these mice that were bearing bone tumors in the bones had much better survival as a result of blocking the TGF-beta in combination with immune checkpoint therapy. And so now there are clinical trials that are being proposed for this combination, and we're waiting for the anti-TGF-beta phase 1 safety data before we can move forward with a combination therapy study. I want to talk a little bit about toxicities because, as I mentioned, although we had these responses that occurred, the clinical trial did not continue because toxicities limited the continuation of combination therapy in these patients. And so toxicities is very different with immune checkpoint therapy as compared to other chemotherapy or radiation therapy. And the majority of toxicities are inflammatory, right? They have this ITIS ending, colitis, pneumonitis, hepatitis, hypophysitis. And what we've noticed is that colitis may be very frequent in patients who have immune checkpoint therapy. Maybe 5-10% of patients can develop colitis, but they're very manageable. And so the fatality that occurs from colitis is minimal compared to what happens when you have myositis or myocarditis. And so these were very prominent in some of the studies, and we were starting to think about these because, again, even though they rarely occur, as you can see here, maybe less than 1% of patients develop them, they have a high fatality rate associated with them. Excuse me. So we decided to collect samples from patients. And so we undertook a study whereby we could collect samples, meaning the patients had cardiac biopsies and skeletal muscle biopsies from our clinic. And these studies were done in collaboration with Bilal Siddiqui, Sumit Subudi, and Nick Palaskis at MD Anderson. And you can see now we were able to collect these samples from patients and perform single-cell RNA sequencing from confirmed toxicities to 11 patients and control patients who received immune checkpoint therapy but did not have any confirmed toxicities when they presented. And we had cardiac muscle biopsies and skeletal muscle biopsies for single-cell RNA sequencing and immunohistochemical studies. And what we found was that when we compared normal cardiac tissues to myocarditis, and I want you to look at the plot here on the UMAP, we found this significant increase in T cells in the cardiac muscles. Same thing in the skeletal muscle. You can see now that in the T cell subset here, there's a significant increase. Similarly, for myeloid cells, you can see here in the normal cardiac muscle, the myeloid cells are very few, but here in the myocarditis setting, we see this increase, and same thing for the skeletal setting, this increase in myositis in the myeloid cells. So we did in-depth analyses to review these cell subsets more closely. And what we found was that there was this increase in IL-1 beta positive, TNF alpha positive myeloid cells that really went correlated with the increased myocarditis that we were seeing in our patients. And so now there's an agent that is an IL-1 pathway antagonist. And so this agent is now being proposed as a new clinical trial to treat patients with immune checkpoint therapy induced myocarditis. So we hope to see that start soon as a result of these data that we generated. So what happens when we think about all of these different myeloid subsets that I showed you, these immunosuppressive subsets, VISTA positive cells, PD-L1 positive myeloid cells, IL-1 beta positive myeloid cells? Are we going to try and target all of these immunosuppressive subsets with different single agents and then hope that the combination of all of these agents are not too toxic for our patients? Or are there upstream epigenetic pathways that can be targeted to reprogram immunosuppressive myeloid cells? And I just want to highlight one work that was done with Sankita Goswami, who was in my lab and now has her own independent, wonderful lab. But she published this paper in 2023 looking at what are the epigenetic pathways that define immunosuppressive myeloid cells in an immune checkpoint therapy resistant tumor such as glioblastoma. And here you can see she looked at multiple epigenetic pathways to really understand which one is expressed in all of the different immune suppressive myeloid cells. So these clusters that you're looking at are clusters that are defining the immunosuppressive myeloid cells. And then we wanted to see, is there a single epigenetic pathway that may define them? And what we highlighted here was that KDM6B did seem to cover most of the immunosuppressive myeloid cells in this tumor setting. And so we did experiments then in a lysim-Cree KDM6B flox mice, and this allows us to remove KDM6B just in myeloid cells and ask what happens when KDM6B is removed in these myeloid cells. How does it change the immune response? And when we did cytoph analyses on these tumor samples from these mice, or where we took GBM, injected it into the brains of the mice, you can see when you looked in the KDM6B flox mice, now the myeloid cells were decreasing in terms of TGF beta immunosuppressive myeloid cells, and multiple other immunosuppressive myeloid cells were decreasing. And this allowed for now an increase in CD8 T effector cells within the tumor microenvironment. And so these mice had much better survival compared to their wild type counterparts. So it was very interesting that there is a way to epigenetically regulate these cells. And so this led us also to wonder, are there upstream elements that control differentiation and function of T cell subsets? Are there other epigenetic pathways also for T cells? And as I mentioned, there's this yin and yang to the immune response, whereby if you target CD28 and T cell receptor, you're going to turn on the T cells. But what we've known for some time is that you also increase this EZH2 expression. And EZH2 is this epigenetic program that allows for stabilization of the FOXP3 transcription factor. And so this allows effector T cells to become more regulatory-like in a way to control the immune response. And so when we looked in patients who had previously received anti-CTLA-4 therapy, we looked at their CD4 T cells. And what we found in that patients who were not responding as well to immune checkpoint therapy, they were increasing this EZH2 expression. And so we wondered whether or not we can then look at EZH2 more carefully in order to consider inhibiting EZH2 as a combination study. And so to do these studies, we looked at FOXP3 GFP mice, whereby we can then sort the FOXP3 positive cells by GFP expression. And then we can incubate these cells with or without an EZH2 inhibitor and perform RNA sequencing and conduct functional studies to understand whether we phenotypically and functionally change these cells as a result of EZH2 inhibition. And what we were able to show is that the EZH2 inhibitor did decrease FOXP3 expression and also increase interferon gamma related genes within these T cells. So phenotypically, we were definitely changing them. Functionally, when we did the co-culture assay, whereby we asked if effector cells can proliferate with regulatory T cells from these studies, we can show that without the EZH2 inhibitor, these regulatory T cells are very good at suppressing effector T cell proliferation. But once we have the EZH2 inhibitor on board, these regulatory T cells are no longer capable of functioning as regulatory T cells, and therefore effector T cells are now quite capable of proliferating, indicating again the ability to suppress these regulatory T cells from functioning with an EZH2 inhibitor. So then we asked whether or not we can do this in a combination study in a mouse model. And here I'm showing that when we combined an EZH2 inhibitor with anti-CTLA-4, you can see the tumor volume significantly decreases, as shown here in this orange line. and survival significantly improves as a result of combination therapy. And so we took these data from our mouse studies and we decided to conduct a new clinical trial. And so this is just showing you how we take the information that we learn in patients about EZH2 being increased. We go back to the laboratory to study what are the mechanisms that can be involved in testing preclinical models. And then we come back to the clinic again in this iterative process to test whether or not this new combination can be meaningful for our patients. And here, this is a clinical trial with two companies. Daiichi was the company that had the EZH1/2 inhibitor, and Bristol Myers Squibb had ipilimumab. And we asked these two companies to work with us to then do this combination clinical trial. And you can see this clinical trial is still ongoing. We're accruing patients with prostate cancer, bladder cancer, and renal cell carcinoma. And in the patients with bladder and renal cell carcinoma, since anti-PD-1 is already an FDA-approved therapy for those patients, these patients can enroll in our trial even after their disease has progressed on anti-PD-1 therapy. And again, these are all GU medical oncology tumor types, and so they're treated in my clinic, and Sangeeta is able to accrue them also in her clinic. So it's really a good way for us to test our hypothesis about the combination in patients. We're also collecting all of the tumor samples throughout time, as shown here, so that we can really understand what's happening within the tumor microenvironment after we give the combination treatment. And so the clinical trial is still ongoing, but I do want to share some preliminary data because we're very excited to see that even in patients who had prior anti-PD-1 therapy and they've progressed through their treatment, now on this new clinical trial, you can see on the CT scan, the post-treatment scan shows that the patient is responding to treatment, which is great. Now, I hope we have more patients like that. So it's still too early to make any definitive statements. But what we're happy to see is that if you look at this cluster 3 on the single cell RNA sequencing data set, you can see the pre-treatment sample has very little of this cluster 3, which are these effector CD8 granzyme K positive interferon gamma positive cells. But after treatment with the combination, now you can see this increase in this effector T cell subset. And we also saw a decrease in the myeloid subsets, which is, again, something that we've been able to show over and over, is that once we increase these effector sets and decrease the suppressive subsets, we tend to get a much better anti-tumor response. And so the clinical trial is ongoing, and I hope to share updates at a later time with you as we complete the trial. I do want to highlight that all of the work I've shown today is really a team effort. This is a team effort where the immunotherapy platform is able to move very quickly between clinical trials and laboratory interrogation. Many, many, many people from many, many different teams involved here. We have about 111 trials where we're doing this across 18 departments in MD Anderson, and about 4,000, actually it's up to 5,000 patients enrolled to date, and multiple longitudinal samples, as you can see, that we're interrogating. So I think it's very important to continue to do these kinds of studies so that we can really understand which targets to go after in larger clinical trials. So in conclusion, I just want to say immune checkpoint therapy has joined the ranks of surgery, radiation, chemotherapy as a pillar of cancer treatment. Combination strategies are the future, but we have to understand which combination to give for which patient. Multiple immune checkpoints exist. They're very dynamic in their expression, and they have to be evaluated in both pre and on treatment human tumor samples to guide therapeutic decisions. We cannot ever use a single time point for understanding human immune responses. The organ-specific microenvironment will need to be considered, since I showed you that there's a difference between soft tissue metastases versus bone metastases. Epigenetic regulation of immune cell subsets is a feasible strategy for combination with immune checkpoint therapy to improve anti-tumor immune responses. The potential overlap between immune cell subsets that drive anti-tumor responses and toxicities, which will require identification of specific pathways, possibly related to myeloid cells, that can be targeted to minimize toxicities while maintaining or improving efficacy. This is important because I think people are trying to dissect these, and what we're seeing is that some of these subsets really do overlap with having an anti-tumor response and toxicities. And then I hope I've convinced you that pre-surgical or these neoadjuvant and tissue-based clinical trials provide a feasible platform for us to obtain approval of treatment for patients with earlier disease, not just stage 4 disease, but also to study biologic effects in patients, which provide insights into mechanisms that can be targeted for rational combination therapies. I have many, many people to thank. I mentioned them as we were going, and their names were on the slide, but I especially want to thank our patients who continue to participate in our clinical trials. So thank you, and I'm happy to take any questions.
Thank you very much, Pam. Thanks so much for this great talk, for the wonderful work and this very exciting adventure. from basic science to clinical trials, and from clinical trials to basic science again. I think this is showing us how is the road to go, the way to go. So thanks so much. And as usual, this talk will be followed by questions and answers in Twitter X at Global Immuno, hashtag Global Immuno. So these questions will be forwarded to Dr. Sharma. Dr. Sharma will be able to answer them, and we will be back. So finally, I would like to announce that our next speaker next week will be Michaela Gag. So thank you very much to all the audience. Thanks again, Pam. We are really grateful with you.
Thank you so much. Take care, everyone. Bye-bye.
Take care. Thanks.